From now on, every time you see a protein structure in one of these, what we call a ribbon diagram representation, where you have these alpha helices and then you have the beta strands as these flat arrows, think this amazing scientist, Jane Richardson, who I want to tell you a little more about, and also more about these ribbon diagrams, which help us visualize what's going on in a protein structure. So I talked yesterday about X-ray crystallography, and I'm not going to go into the details here, but you start with this beautiful protein crystal, and you end up with some like beautiful structure like this, right? There's a couple of confusing looking steps in between, because what you get is like this indirect evidence that this like pattern of spots from, so you shoot x-rays at the crystal, and then the atoms in the crystal interact with those x-rays, and then they diffract, so they like scatter the x-rays, and those x-rays interact and they give you this pattern of spots called the diffraction pattern, and then you work backwards from the pattern to get the electron density map, which is something not so pretty and really hard to make out what's going on. So scientists have methods that they can build the model into that mesh. So locate the positions of the fit the positions of the various atoms into that mesh, which represents the positions of like the electrons around the atoms. And so now we don't need that mesh. We can, if we remove the mesh, you can see that it's still kind of hard to make out what's going on, right? Even though I have it colored pretty, which is obviously an artistic representation. But it's still kind of hard to make out what's going on. But when you make it in this ribbon diagram form, now you can clearly see the various elements of the protein backbone and the different motifs that they make. So we call those like coils and strands, so like secondary structure motifs that involve the backbone of the protein. I'll tell you more about that in a minute. But the basic point is that now it's easier to see what's going on in various regions of the protein, and to see the overall structure of the protein in this much easier to interpret way. And we have Jane Richardson to thank for these diagram structures that you see like all the time. So I want to tell you more about her and more about these ribbon diagrams and about the various ways that we can display proteins. I had the pleasure of meeting Jane Richardson a couple of times at Cold Spring Harbor when she'd come to help teach the crystallography course. And she is just like an amazing woman. So more in this post about her, but she's like this Renaissance woman. She studied like space stuff and all sorts of like philosophy, biophysics, astronomy, and computational biology. But she doesn't even have a PhD. She's just incredible. She's like mainly self-taught. She worked with her husband, David Richardson. They saw some of the first protein structures, so figured out like the atomic makeup of like where all the different atoms were positioned. And they were getting all of this data and getting this big jumbledy looking things, and she wanted a way to really represent what was going on in a way that was clearer to interpret than things like just having like a ton of sticks. Like, how do you actually like interpret what's what going on in here? And even the sticks is better than just like the if you just had the map. So you just had this giant meshy thing or whatever. So basically scientists needed a way to be able to represent what was going on in the structure in a way that was more easily interpretable, and it'd help if it was like pretty, right? But anyway, so she would actually work with like making physical models of like wrapping ribbons around tubes and all of this stuff. It was really incredible to hear about. And so she would start with like with her drawings and stuff, making the carbon alpha backbone, so the backbone of the protein. So basically proteins are these long strands of amino acids, which are like individual letters, and then they fold up into these complex shapes. And she realized some important things, like that the shapes, there's importance. She focused a lot on the secondary structure, so that's the interactions that involve the backbone of the protein, and they can form these common motifs. So these like beta strands, So these long jaggedy strands that you'll see, like these things. So these are beta strands, and then you can have strands next to each other to form these like beta pleated sheets. And so you can have these parallel or anti-parallel. And so to represent the direction that they're running, she drew them as these arrows. So you'll see that the arrows go the same direction if they're parallel. Sorry, this one should be, yeah. So these two structures should be opposite. So these ones are parallel and these are anti-parallel. And so she represented these beta strands as these arrows, and then the alpha helices as these spirals, these ribbons. And this really helped turn these crazy looking things into something that was really easy to interpret what was going on in the structure at the basic level. So of course you lose a lot of the detail when you do this. You can't see all of like the individual side chains, so all of like the unique parts of those amino acids that stick out. But what happens is that in crystallography, what actually the strongest signal that you're going to get is from that backbone, because the positions of those side chains can be like more floppy or whatever. The important thing that Jane Richardson, well, one of the important things that she taught us is that there's no such thing as photorealism when it comes to proteins. So I think of proteins as these like gorgeous things, but these things are so tiny that like this is not like what a protein, like, it's hard to explain, but we can't really wrap our minds around what a protein would like actually look like because we can't see it with our eyes. So there's no like photorealism, so we can display it in these various ways. So we're still representing the position of the atoms, but we're showing it in different representations because we're not seeing it like directly with our eyes. And the crazy thing with crystallography is we're not even like we're seeing like indirect evidence of it and working backwards. So what we're seeing here, like all these beautiful ribbon diagrams, these are like models, and so the actual data that we have comes from that pattern of diffraction spots, and that pattern of diffraction spots is how we build these electron density maps, and then we build the model into those maps. So it's really this like convoluted way of getting to this structure, but the structure ends up being something so beautiful. You can see like an alpha helix, and you can see those beta strands, and then the beta pleated sheets where you have strands next to each other, and all of these things. And so you'll see too that some regions have a lot better density than others, and so you can almost always see the continuous backbone, which is the strongest part and stuff, and that's what's really well represented in ribbon diagrams, thanks to Jane Richardson, who gave us this way to represent these structures with these ribbon diagrams. Another important thing that she's been involved in is developing this MolProbity software, and it helps point out potential problems in structures. And so it's actually like integrated into various structure building tools like Phoenix or Find, which is really helpful. But basically it points out problems areas in a structure. So if there's like atoms and positions that clash, and weird angles and that sort of thing. So basically when we're looking at the map, you're not just trying to get the model, like if you were to just rely on trying to match the shapes into the mesh, it would be like really difficult and potentially not that helpful, because remember that the mesh is going to the mesh can improve as you improve your model. And so because you're improving the phase information, which you don't have. So you want to rely on prior knowledge such as about the general geometry and the idea of like steric clashing and hindrance, and that you can't have two molecules occupying the exact two atoms occupying the same space, or they clash and that sort of thing. And so the MolProbity software helps you find areas where the protein is kind of like defying the laws of physics and biochemistry and that sort of thing. So places that you should be improving your model. And so she makes it really visual once again, and to really point out these differences with things like these clashing symbols and that sort of thing, and really integrating it into the software so that it makes it easier to find and fix these mistakes. So she works closely with her husband David in their lab at Duke, and she's also really involved with she told me like she done a lot of work with like Wikipedia editing, as well as putting a bunch of resources and drawings and stuff onto Wikimedia Commons so anyone could use it. So she's this really amazing person. She's also been involved in the coronavirus structural biology task force, so I'm trying to make sure that the structures of all of the coronavirus proteins in the data bank and stuff have really good models and that sort of thing. And so she's a really big inspiration of mine. So I've been thinking about her a lot.